Hello everyone, welcome back to my channel. In this session, I'll be discussing about a very important topic, and that is Griffith's experiment, which can also be called as transforming principle. Before I start the session, do not forget to like, share, comment and subscribe. So let us start the session with an introduction. Fredrick Griffith conducted a lot of series of experiment in which he had taken Streptococcus pneumonia, which is a bacteria, and the animal mice as a model, and this experiment was conducted in the year 1928. A very important point to be noted here, Fredrick Griffith concluded that the R strain bacteria picked up a very important principle which is called as transforming principle from heat killed S strain bacteria, which converted the R strain bacteria into smooth coated bacteria and virulent bacteria. Here virulent means it will be pathogenic, which causes diseases. Now let us know who is Fredrick Griffith. Fredrick Griffith is an English bacteriologist who started his experiment in 1928, which was named as Griffith's experiment. Griffith was first person to discover the principle which is called as transforming principle. The transforming principle also led to the discovery that DNA, which is an deoxyribonucleic acid, as a carrier of genetic information. That means this genetic material could carry the certain information or particular information from one organism to the another. He also concluded that through his discovery, bacteria can transfer genetic information via a process which is called as transformation. That is why this principle is called as transforming principle. And Griffith's work was expanded by Avery, MacLeod and McCarthy. Now let us learn about the Fredrick Griffith experiment. Fredrick Griffith had taken two different Streptococcus pneumonia strains, which were called as R strain and S strain bacteria. Streptococcus pneumonia is a bacteria which comes in several types and strains. We can either call it as types, we can either call it as strains. Griffith chose two different strains for his experiment, and the first one was S strain bacteria. The second one was R strain bacteria, as you can see on the screen. S strain bacteria, which is called as smooth strain bacteria, because this bacteria had a smooth surface or a protective covering which was made up of polysaccharide coat. That is why it was called as smooth strain bacteria, and these bacteria were virulent or pathogenic bacteria which could cause diseases. Why? Because they had the polysaccharide coat which was covering them and they were virulent. The second type is R strain bacteria, which is called as rough strain bacteria. This is because the outer covering was rough, it was not smooth. That is why it's called as rough strain bacteria. And since there was absence of polysaccharide coat, that is why they are called as non-virulent bacteria or non-pathogenic bacteria, which could not cause any kind of diseases. Also a very important point to be noted, Fredrick Griffith had taken an animal model, that is mice. So when he injected S strain bacteria, which is a virulent bacteria, death occurs in the mice. Wherein when R strain bacteria was injected into the mice, since it is non-virulent bacteria, death in the mice was not seen. Important point to be noted here, when Fredrick Griffith observed that when S strain bacteria were injected into the mice died because of pneumonia. Wherein R strain bacteria were injected into the mice survived. Now let us study through the diagram. Now I'll be explaining about Griffith's transformation experiment Already we have learned that he had taken two different types of strains, R strain and S strain. But before that, why he had conducted this experiment? He wanted to research the possibility of developing a vaccine, that is a pneumonia vaccine. So when he started, he found out these two strains which were from the Streptococcus pneumonia, which is a bacteria again that infect the mice. The first one was already I have told you previously, S strain is a smooth strain. And why it's called as smooth strain? Because it is having a covering or a protective layer or a capsule which can protect itself from the host immune system, and that is why it's called as a virulent type or a disease causing type of bacteria, isn't it? And the second one will be R strain, and that is also called as the rough strain, and it is a non-virulent type of bacteria. Why? Because there is an absence of the protective capsule, plus it does not cause any kind of diseases. That is why it's called as non-violent or non-pathogenic type of bacteria.
Fredrick Griffith injects the rough strain, which is a non-virulent, into the mouse and the mouse lives. Then he takes the smooth strain bacteria, which is virulent, and injects into the mice. The mouse dies because of pneumonia. In the third attempt, Griffith attempted to inject mice with heat-kill S strain bacteria as a part of his research. That is, S strain bacteria had been heated to high temperature, causing the cells to die. But unsurprisingly, the mice lives.
Coming to the last step in the Griffith's experiment, as you can see on the screen, that when the harmless R strain bacteria were mixed with harmless heat-killed S strain bacteria, which was injected into the mice developed pneumonia and died. But how? Griffith observed that the S strain bacteria was present in the blood sample taken from the dead mouse. So in this experiment, what did he conclude? He concluded that some factor or biomolecule, that is mainly from the heat-killed S strain, entered into the R strain, okay, to synthesize a polysaccharide coat and become a virulent bacteria. This is why he calls this experiment as a transforming principle. Because of a factor, an S bacteria can enter into R bacteria, and R bacteria can get completely transformed into a virulent bacteria. This is the transforming principle. This process is now known as bacterial transformation, and it is significantly used in genetic engineering applications.
Conclusion: Griffith's ultimate goal was to find a way to cure pneumonia. Griffith concluded that heat-killed virulent bacteria transformed living non-virulent bacteria into virulent bacteria. He performed his experiment on the two strains of Streptococcus pneumonia, which differ from each other due to the presence of a polysaccharide coat. Griffith's findings were published in the Journal of Hygiene. In 1928, his experiments with mice led to his major discovery of bacterial transformation. Griffith's experiment discovered that bacteria can transfer genetic information through transformation.
Say what you wanna say to me